And this is looking at the same thing with onion root tip cells. And so we have interphase, so we don't have much of a condensing down of the chromosomes. We have prophase or early prophase, where the chromosomes are becoming condensed down. We have later prophase or prometaphase, where not only are the chromosomes condensed down more, but you can see that they're no longer constrained in the same way within the nucleus as they were previously. We have further condensing and movement of the chromosomes until the chromosomes are found on the metaphase plate. And that's metaphase right here. And then we have separation of the chromosomes, which I'm not convinced this is actually anaphase here. This is probably early telophase here. I was not able to find a good anaphase. We'll see that in a second. And this is reformation of the nucleus as telophase. So this actually is based upon photographs that I took through our microscopes, admittedly using oil, but through our microscopes yesterday, and taking pictures through the ocular lens using my digital camera. And then I just played with them in Picasa and pasted them all together and turned them into this. And although that looks moderately okay, it really is a good representation of what you see, although it's much bigger than what you'll actually see. This is what really came out nicely. So this is the whitefish blastula cells that we have in lab. And what we have here is interphase. What we have here is prophase. And you can see that you've got the asters sitting up here. It's amazing you can actually see them. You have here further compacting down of the chromosomes in prophase phase. Here we have metaphase. So they're clearly lined up. You can clearly see the asters. So the centrosomes are found here. We'll have the kinetochore microtubules that are connected to the chromosomes, the non-kinetic cores pushing apart the cells. And then we have here what clearly is anaphase, and I put it there, although it's a later anaphase, and I put it there twice or even 3 times to make up for the fact that in the previous slide it didn't have any anaphase. And then they reach the end. This is presumably telophase. You now have, they've reached where they're going to end up. You actually seemingly have the beginning of cytokinesis, and then you have the reformation of the nuclei. Here we don't seem to have any division in the cytoplasm, so I'm calling that telophase. And then here we actually have reached the point where it would appear that those are 2 daughter cells of a single cell division, if only because they look very similar to one another. So they're still maybe in late telophase. But this is the whitefish blastula cells, and this literally was taken from the slides we use in the classroom, using the microscopes we use in the classroom. Again, I put these under oil. But you can see this because I was trying to blow them up to make them big. But you can see more or less this kind of detail in those slides. So mitosis in an animal cell.